AHRI pooling resources to put out a consistent message about managing herbicide-resistant weeds. And yeah, this webinar series has been part of the Weed Smart initiative. This morning we've got Peter Boutsalis on the line. Good morning, Peter.
Good morning, everyone.
We'll introduce Peter in a minute, but just while a few more people are logging on, we'll just do a little quick minute of housekeeping, and then we'll hand over to Peter. Excuse me, everyone. I've got a cough, so hopefully I don't cough down the microphone too much. Just that bit of housekeeping. You've got a menu box probably on the right-hand side of your screen there. It might be a good idea to minimise that by clicking on that orange arrow so it's out of your way. But if you'd like to type a question in, and it's very good to have lots of questions come in, then please maximise your box there and type in a question into the question or chat box there, and we'll address questions as we go. So as we go, in Peter's presentation, this sort of slide will come up periodically, and when that happens, we're going to pause and do some questions and discussion, and then keep on going, because I'm sitting in Western Australia and Peter's sitting in South Australia, so we're going to give that a go to try and keep the questions coming through. Just if you have some problems, probably the best thing to do is write down Kate's phone number there, Kate's mobile number. If you drop off the webinar for any particular reason, give Kate a call and hopefully she can fix your problems. You can write down the webinar ID there and go back to that web address and rejoin. But if it was me, I think I'd be bugging Kate. And thanks again to Kate from Coccinall. Kate is just about to go on maternity leave with her first child, and good luck with that, Kate. It's been great to have her in the co-pilot seat, and yeah, we've really enjoyed having you organise these webinars. It's made it very easy for us. Okay, so probably time to get into it. We've got 66 people out there logged on, and so it's a good audience, and we'll be going for at maximum an hour, but we'll just see how wound up Peter Boutsalis gets, I suppose. So introducing Peter Boutsalis, he did his PhD at the University of Adelaide, moved to Europe, working in Switzerland and England with Syngenta for 8 years, then moved back to a research position at the University of Adelaide, where he is now. And Peter also runs Plant Science Consulting, which many of you would know offers a herbicide resistance testing service, and he can tell you more about that. But it's not only his research, but it's his looking at all of the research, all the resistance test results between clethodim and factor, which has given him an insight today. So the reason we wanted to do this topic is that with this time of year, we're sort of focusing on the post-emergent herbicides, and we know that we've got a lot of clethodim resistance. We know that butroxydim or factor is an option. We just want to tap into Peter's knowledge to find out when it may be when are the best times to switch from select to factor. So I'll hand over to you there, Peter, and then we'll have a bit of discussion and questions as we go.
Okay, good morning, everybody. It's nice and wet in Adelaide this morning, so good start. Okay. So when is it worth rotating from select? We'll use select. There's a lot of herbicides available with clethodim to butroxydim. Okay, now I'm going to do a little bit of history so we all understand how the group A's operate and how they're classed. So as all of you would know, they're classed according to, they're all group A's. We've got FOPs, there's several herbicides in the FOPs. We've got DIMs, there's fewer DIMs, and we've got an AHRI, which is a den herbicide. It's a different chemical altogether. So that's classifying them according to their chemical structure. Now I'd like to class them also according to their selectivity to wheat. So we've got group A's that are selective in wheat, and we've got others that are not. So where you've got group A selective herbicides in wheat, resistance can be due to metabolic or target site resistance, and we'll go through these. So they're herbicides like Ryegrass, Topic, Achieve, AHRI, and there's a few others too, like your Cheetah Golds, and Decision is another one. So resistance to those can be due to target site resistance or metabolic, or as happens also, both mechanisms in the same individual plant. Broadleaf, you've got broadleaf selective herbicides as well. These are herbicides which kill wheat. They kill wheat because they can't be metabolized in wheat. Whereas things like Ryegrass and Topic achieve axial, wheat actually metabolizes them into non-toxic compounds, whereas susceptible ryegrass cannot, and that's why they kill those weeds. Whereas the broadleaf crop selective group A's, they can't be metabolized in wheat. And generally what we find, if plants are resistant to these herbicides, so you've got ryegrass resistant to verdict, target, select or factor, that's almost always due to target site resistance. Okay, so if you see any plants resistant to those herbicides, think if you had a bet on something, it would be target site resistance would be the culprit. Okay, moving to the next slide. Now, how do the DIMs work, or all group A's? They all inhibit to a single enzyme. This enzyme is acyl coenzyme A carboxylase, or ACCase for short. So one enzyme. They all bind to one site on that enzyme, but the binding is not identical, so they bind slightly differently in that pocket. So where you get changes occurring at this binding site, and we call these changes mutations, then herbicides start to not recognize or don't bind as strongly or don't bind at all, and therefore if the herbicide is not binding to that site of action, then the enzyme continues to work and the plant survives. So that's the crux of how these guys work. So just a little demonstration, because we've got variations even within a chemical group, like say the DIMs, where we've got achieve, select, factor. We used to have the herbicide clethodim or certain, that was available, but not available anymore. These herbicides still bind slightly differently to the enzyme. So here we have just an example of the ACCase enzyme, the target enzyme, and we have a binding pocket there. So when a herbicide binds, it inactivates that target enzyme and the plant basically starves to death. Now we can spray another DIM herbicide, and both of those are binding, inactivating that pocket, the enzyme, but they're binding, as you can see by the different colors, not the same. So that's a susceptible enzyme. Now here we have a resistant enzyme. It's resistant because if you look at that pocket, there's another yellow component with black spots. That's we just picture by picture cartoon, we're representing a mutation. So when you spray herbicide 2, it kills the plant, whereas if you spray herbicide 1, it doesn't recognize. So that could be a situation where herbicide 2 is clethodim. Clethodim will kill the ryegrass, but if you spray achieve won't. And we have lots of cases where clethodim is active on ryegrass, whereas achieve is not. Both DIMs, but very different effect. But you can also have mutations in other sites where now you get herbicide 1 working, but not herbicide 2, because the mutation is in a different location on the same binding pocket. That's how we explain in simple terms how differences in mutations can cause different resistance to herbicide in the same mode of action class. Oops, sorry. Okay. So the ACCase gene is actually made up of 4 components, and this is a small segment of one of those components, and that is where we find most of these mutations occurring. So this component here is the actual binding site, that pocket that was in the previous slide, where the group A's bind. Now you can see there 7 locations starting with 1781. They are physical regions where if a mutation occurs at one of those sites, it will give group A target site resistance to some of those or all of those group A herbicides. There's 7 locations, 7 mutations. Now different, so you get difference occurring. At some sites, you can also get like say at the 1781, you can also get more than one mutation occurring there, and if that occurs, then you get different resistance profiles. So not only do you have 7 potential areas on that binding pocket where a mutation will cause resistance, you can actually get different mutations occurring at one of those sites, which also gives different resistance. So you can see it can get quite complex, and that's why you can get, you know, varying degrees of resistance to different group A's. What we find is that the mutations, most of these mutations, give resistance to FOPs easier than they give it to DIMs as well. Now what we'll find later on in the presentation is that some ryegrass have more than one mutation, and when that occurs, you start to get to that scary levels of group A resistance. But that is the actual pocket, the DNA sequence of that pocket, which causes those mutations. So here's some work that was done, published in a paper, where on the left, the amino acid substitution, they've actually got those positions that we saw in the previous slide. So we've got the 1781, the 1999, 2027, etc. And the APP, they're FOP herbicides. That's the international code for FOP herbicides. And CHD are the DIM herbicides. So you can see that the 1781 isoleucine, ILE to leucine, gives you resistance to the FOPs and also resistance to the DIMs. Now this table here is not only for ryegrass, it's also for some grasses that occur overseas, such as blackgrass. There's another one called Setaria foxtails. There's also a few more. So the profiles aren't exactly as we see in ryegrass, but they're quite similar. But this isoleucine to leucine 1781 gives you more DIM resistance than FOP resistance, and because it's got 3 stars versus 3 crosses versus 2 crosses. We don't see that in ryegrass, but we actually have seen that in some wild oat populations in Australia. The next mutation, the TRP to cysteine, the 1999, gives you strong FOP resistance, but no DIM resistance, and that is quite, we see that a lot in ryegrass as well. The one after, actually the next 3 mutations will give you stronger FOP resistance than DIM resistance, and we see that in ryegrass. And yeah, the final one also, actually they will give you that. The 2088 one will give you equal FOP and DIM resistance. So you can see that those mutations are giving different resistance profiles, but generally they give you stronger FOP resistance than DIM resistance, particularly in ryegrass. Okay, so that's the first little section, and I think Pete's going to see if there's any questions for me.
Yeah, well, just keep your questions coming in, everyone. We haven't got any in there yet, but if there is a question, make sure you type it in, and that's the beauty of these webinars. I guess for me, Peter, the interesting thing, and it's a realisation I came to recently as well, is any of these herbicides that, you know, any herbicide that's a group A and it's tolerated by wheat means that you can get metabolic resistance to that herbicide, because that's how the wheat tolerates it, isn't it? Whereas
exactly right.
any herbicide that's not tolerated by wheat, you don't get metabolic resistance, just target side. That's it, isn't it?
That's right. Yep, wheat has a different metabolic pathway than ryegrass, but the ryegrass that's resistant due to metabolic has adopted the wheat type metabolism.
Yeah. Okay, I've got a couple of questions come in. There was one question saying there is some talk that people are getting diclofop to work again where they haven't used it for 10 to 20 years, even though they've been using other group A's. Can we expect susceptibility to come back to life even though we've been using the DIMs and DENs? So I'm not sure if that's true. Peter, you might know better. I'm not sure if anyone's actually got diclofop to work again.
No, I had a situation like that where that was quoted, and I tested the seeds in a pot test, and I couldn't see that performance. So I'd be very surprised if we get diclofop working and resistance to, say, your chiefs and axils. That would be very surprising.
Okay. two more questions come up here. We know that there's 7 mutations there, Pete, that you've mentioned, but is it likely that there will be any more, or do you think? I guess the answer to that is yes. But what is the likelihood that we'll find more mutations?
Oh, look, I think there is. We've got a few populations. I think I may have got it later on in the presentation, but there are a few populations which are highly resistant to clethodim and butroxydim. They're like resistant to a liter of clethodim and very high rates of butroxydim. And where we've looked, they don't have any of those 7 mutations. So we're really scratching our head to see, is there an 8th mutation, or is there a new monster mechanism in that ryegrass that's giving that very strong resistance? So yeah, I'm sure that's not the last that we've seen of those.
And we'll just do one more question now, but this is one that I think that you're going to answer later, so if you are, you can do it then. But if Group A is target site, not metabolic, why are we increasing clethodim rates? And this is the million-dollar question. So, do you want to tackle that now, Peter, or do you want to leave that to
I'll explain that, and if I haven't covered it, then I'll definitely go to that question. But yeah, majority of the presentation is to answer that question of why increasing rates work.
Yep. Okay. There's another question there. We might leave it for a minute and we'll let you keep going, and then we'll come back to more questions. So yeah, keep the questions coming in, everyone, and we'll stop periodically and cover them. So okay, launch away, Pete.
Okay, this is a situation where I would suspect resistance, and it's not, it's not always as clear. I've been in paddocks where I've looked at weeds surviving and I've thought, yep, absolutely resistant, and where we've taken plants and done a quick test on them, they've all died. So it's not always the same. But here we've got a patch in a canola crop which has got dead brome but ryegrass surviving. Now that first tells me that that herbicide has actually worked. It's probably I'd say a select verdict mix. So that there's been good herbicide activity, but that ryegrass is surviving. Even though, if you look carefully, and I'll start to move my mouse and go towards the back, there's a ryegrass here that's dead or dying. So there's some differences there, but that situation just starts to tell me that the herbicides have worked, but there's something that ryegrass is surviving, most likely due to Group A resistance. The next slide has a little bit more information, and that is where we've got ryegrass. I'll move the mouse, that is alive. Ryegrass that's inhibited but is producing new shoots, and we've also got dead ryegrass. So we've got all, from basically very healthy ryegrass to dead ryegrass to everything in between. And here, my brain starts to think, hey, this has been sprayed with clethodim, so anything that's surviving there is target site resistant, but there's differences in the recovery, and so that's indicating that there's some different mutations occurring even in that little patch. Okay, so that's a kind of information that you can extract from that. And also you've had good weed control because there are some dead plants there, so the herbicides have worked, and so those surviving are there for another reason. So to answer that question and that last question, we've actually got some hard data of the distribution of Group A herbicide resistance in a paddock, and why higher rates of clethodim and butroxydim are required these days in many paddocks to actually control ryegrass, where earlier 250 mils of clethodim would have been more than enough. So what we have here is some work that we did at Adelaide Uni a while back. Actually, I wasn't here when they did that work, but this is where they went into 3 paddocks, and every 10 meters stopped and picked an individual ryegrass plant and did that for the entire 3 paddocks and kept them all separate. So the seed was cleaned. That was done by my wife. She wasn't happy that summer, but she cleaned the seed, and then we tested each plant individually with, I believe it was fusilade we tested it with. So one of those herbicides that gives you, that are selective only in broadleaf crops. And we found various survivors. Now, each of those spots indicates a ryegrass that survived fusillade. The different colors represent different group A target site mutations. So you see there's a lot of red there. There's 25 red spots. That is, we found 25 plants that had that 2078 mutation. 3 plants had the 2041 mutation. Now here's where ryegrass starts to get cunning. Ryegrass is an obligate outcrosser. That means it must receive pollen from another plant in order to produce fertile seed. So if you take a single ryegrass plant in a pot and put it in your lounge room, I'm sure your wife won't be happy with that, but put it in your lounge room and you ensure that no other pollen gets in that room, that ryegrass will produce a lot of pollen but no seed. So it must obtain pollen from another source. So what is happening that if you've got a 2078 plant, a plant with a 2078 mutation next to a plant with a 2041 mutation, they're going to cross-pollinate. And so the progeny, some of that progeny will have both mutations. When that starts to happen, then plants start surviving higher rates of clethodim. If you move down, you've got 5 plants with a 2078 and a 2088 mutation. We didn't actually find any plants with an individual 2088 mutation, but here there's 5 plants with both. There's 1 plant with a 2041 and a 1781, and 1 plant that has 3 mutations and another one that has 3 mutations down the bottom. Now these plants that start to get 2 and 3 mutations, they're more rare. As you can see, there's not many plants in that paddock, but they are the ones that survive very high rates of clethodim and butroxydim. So if you spray that paddock, say, with 500 mils of clethodim, you would most likely kill all those 2078 mutations, so you wouldn't have those 25 plants there, but you'd have about sort of 20 plants surviving in that paddock. They don't look very threatening and you probably wouldn't even see them, but they will be cross-pollinating, accumulating genes and producing seed for next year. And then when it came time to spraying, say, 500 mils of clethodim next time, you'd see more survivors. If you increase the rate further, you'd kill some more of those mutations and only leave the very high ones. And after a while, if they start to dominate the population, then that's where, you know, a liter of clethodim or your full rate of butroxydim just won't touch them. And that's what happens over time when that strong selection. So you can see that, you know, the zero seed, the zero plants, zero seed theory is there because if those plants there's very few plants that aren't impacting yield, if they're the ones that have those complex mutations, they produce seed, they're the ones that are going to dominate the population if they're not kept under control. Some other interesting things from these is you can see that there's 3 paddocks. One paddock has a lot of mutations. The one on the left, paddock 1, has a few, and the one on the right doesn't have any. So here we have 3 paddocks next to each other, and the distribution and the number of resistant individuals is very different. So you can see that even, you know, testing one paddock, the neighbouring paddock could have totally different resistance profile at all. Also, just one other factor is that paddock 1, down the bottom, there's more mutations there, and that is where the gate is. So, you know, those have probably not developed individually, but they've been brought in by either machinery or some other mechanical process. So crop hygiene is really important to try and prevent those resistant guys coming into the paddock, even when the farmers have been doing a very good job of keeping them out. That's the way that they get in. Okay, so I'm sure there'll be a few questions with that slide. I'll just move on to the next one. So that was a paddock level. Now, we've got a Weed Smart, so it's there for any questions.
Yeah, so there's been a few questions come in, not particularly to that slide, but I think it'd be good to have some from that one. So, we'll go with a few questions. One, is there any new chemistry to target different pathways or enzymes? I guess that's yeah.
I can answer that. Yeah, I can answer that question. Yes, there is. We are working with quite a few new chemicals. Part of my job at GRDC is to communicate with chemical companies and for us to look at any promising chemicals. And yep, there are some really exciting new mode of action herbicides that we're working with, and they're targeting, sort of areas which are not existing with herbicides. So yep, there's some really good stuff. Selective in wheat, selective in canola, and probably other crops too. But the main ones we're looking at is wheat and canola, because they're the ones that are mostly under threat.
Mm-hmm. So what are we worried about?
Yeah, look, there are some really good herbicides. One of the things that we have here in South Australia, or we don't have, as my wife said to me, the other day, she was looking at Facebook, is about Roundup Ready canola. We don't have Roundup Ready canola in South Australia, so we're having to contend with controlling dim-resistant ryegrass without the use of glyphosate in crop, and that's making, just a little bit more difficult in some cases.
Yeah. So next question, to what extent could a chemical rate increase lower the potential resistance of a species? So if I'm interpreting this question right, I think it sort of says, what would happen if we had have started with the high rates of clethodim 10 years ago? Would that have slowed down the resistance evolution, do you think, Pete, or do you think it would have happened anyway?
Yeah, look, resistance would have still happened. We would have selected for those stronger mutations. It all boils down to what is done at the end of what sets seed at the end of the season. So yeah, you can spray high rates and you get a lot better kill. You'll still find those rare mutations. If those guys set seed, then the process starts. But it's always best to use the highest rates possible, because then, if you've got fewer plants to start with, you can control them a lot easier in the next season or that season than if you've got, more plants to actually deal with.
I guess it begs a question with this one though. If we, plants accumulate one mutation, don't they? And then if they survive, then they might cross-pollinate with another plant and get two mutations and so on. So if high rates of clethodim kill the ones with a single mutation, could it not have slowed it down because, back in the day we would have killed lots of those ones with a single mutation, and therefore they wouldn't have had the opportunity to gain, 2 and 3 mutations?
Yep. Well, I'm not saying that ryegrass to select high rates of clethodim and butroxydim, I'm not saying that they have to have 2 mutations. I'm pretty sure there are mutations there, single mutations, more rare mutations, that will give, strong resistance to clethodim and butroxydim by themselves. So yeah, it's not only the double mutations which are causing that. They're contributing significantly, but they're not the only ones. So what we don't have as a really good idea is if we list all those possible mutation sites and also the possible mutations, exactly what kind of resistance they cause for ryegrass. That's some serious lab work that needs to be done. So that's a whole different investment situation there.
Okay, so there's quite a few questions coming in, so I'm just going to group a couple together and then we'll keep moving. So there's been questions about other species. So there's been questions about brome grass, barley grass, wild oats. Are we heading down the same path with them? Will resistance evolve as quickly in those species, or will it be slower?
One thing that ryegrass has is it's the obligate outcrosser, so it spreads the pollen. It has to cross-pollinate, whereas those other species that I mentioned, the brome, barley grass, wild oats, they're self-pollinating, and so the resistance occurs much slower. So, okay, if they happen to have a resistance gene, for them to cross-pollinate with another resistant individual and cross-pollinate is very low. So that's why we've seen a lot less resistance in those other species. whereas ryegrass is spreading the resistance around, plus ryegrass numbers are much greater. So there are a few individual components which has caused that in ryegrass. Some of those others also, those other species can have like higher resistance to clethodim than fops, for example. So you can get the reverse. Now we never really see that in ryegrass, but in wild oats we've got situations where the resistance is higher to dims than it is to fops, but we don't see that in ryegrass.
Yeah, so resistance evolution is slower in those other species. It still happens, but it happens at a slower rate.
Yep.
Yeah,
and our random,
go on.
Our GRDC funded random weed surveys have shown that group A resistance in those other species is much lower. So the highest level of group A resistance we have to another species is in Western Victoria, and that's in brome, and that's about 10% of plants we found were resistant. That's the highest. Usually it's well below 10%.
Yeah, similar story in WA.
Yeah.
Now, one last question before we let you keep going, Pete, and that is there any fitness penalty associated with the group A resistance that you've been talking about?
Okay. Some of those mutations do offer a fitness penalty. I couldn't tell you which ones, but there has been some work done by AHRI and others that have shown that some of those group A mutations do offer a fitness penalty, but most of those don't. And so I've tested seed where farmers have said, oh look, last time I sprayed a group A was 10 years ago, could you test it again? And I've tested it and I can still find it in the population, albeit not as high levels as was originally there. So it usually hangs around, but there are, I think some of those double and triple mutations, because there's so much change, they could actually start to impact on the fitness of the plant. So if those herbicides weren't used, maybe those would be weaned out of the population slowly, and so you'd have only either single mutations or no mutations if some of those offer a fitness penalty.
Yeah, there is a bit of fitness penalty, but the plants are generally quite fit when all the other weeds around them die, and then they have less competition. Okay, so we'll let you plug on. I can see there's other questions, but I can also see that I know that you're going to get to some of those just during your presentation, Pete. So we'll let you keep going, and then we'll tackle some more questions in a while.
Okay. So these are the random weed surveys that we've been doing since 2005, and what we've been visiting different areas, and every 5 years we visit the same area, collect weeds at random, and then do lots of pot tests the following year. And at the moment, well, last year we surveyed Eyre Peninsula and Southern Victoria, and we're actually starting to screen those now. But we've got some information. We've had a lot of dealings with that sort of north of Adelaide, because that's close to Adelaide, and that's where when we first started research. So we've actually got 3 survey years of data, and what the girls in the lab did, they did some amazing work where they actually DNA tested and found mutations of individuals from that survey to tell us of those 3 years to see what was happening. Were there any new mutations or what was happening? So this is the next slide here. Oh, this is just an example of a pot test. So each pot there represents a single paddock sample, and as you can see, there's a lot of differences in there. I think that one is more, I think that survey there was Western Victoria, where the upper part, they're in order, so the upper part of that slide, that was the Wimmera, which had more resistance, and that lower part was the Mallee, Western Victorian Mallee, which had less resistance to some of those herbicides. But yeah, moving on. So what we found was that this slide is a little bit complex, but I'll go through it. So this is information from surveys conducted in 1998, 2003, and 2008. So they're 3 years apart, 5 years apart, sorry. So I've got survival data there. So that's the survival in a pot test, and that is the black information. So if we look at 1998 to hoe grass, in 1998, 38% of the ryegrass collected in that survey was resistant to hoe grass. and the only other herbicide tested was clethodim, and that was 19% was resistant to clethodim in 1998. See, if we had sprayed those plants, that 19% with clethodim, we probably would have killed them all, I would say, at that stage. 5 years later, we've got 77% resistance to Ryegrass, so we've had a huge jump in 5-year period, and now we're starting to see some resistance to Achieve, 45%, and Axial, 36%. The interesting thing here was that Axial had not been registered in 2003, so even before registration, 1/3 of the ryegrass in the mid-north was resistant to Axial. Okay, so that just shows you that, you know, what a mode of action resistance can do. And of those, 31% were also resistant to Select at 250 mils. 5 years later, Ryegrass resistance hasn't changed. Resistance to Achieve has gone up sort of 20-fold, it's about the same in Axial, and Select resistance has increased a little bit. So that's what's happened over in 5-year periods. That's weeds selected totally at random. Now, how about the plants with the mutations? So what the girls did is they took the Ryegrass resistant DNA from Ryegrass resistant leaf samples, and they looked to see how many of those had mutations and didn't have mutations. So in 1998, 76% of the Ryegrass resistant had target site resistance, and 24% didn't have group A target site resistance, so they probably had metabolic resistance. As we went through the years, then we've had an increase in the plants having target site resistance and a decrease in the plants having non-target site resistance, which is most likely metabolic resistance. So there's been, over time, there's been an increase in the percentage of plants that have group A mutations. So yeah, I'll leave it there, but the next slide will explain that a little bit more.
So yeah, okay. I don't know if we'll pause now. I think
I think
there's a few more questions, but you.
Yeah, this slide slide is that data that I showed in the previous table. So this has got the 1998, 2003, and 2008 Northern Adelaide surveys. So this is the survey area. Each spot there represents plants, or a single plant from that sample that had survived Ryegrass, and it indicates what kind of mutation it had. So in 1998, we've got 6 plants had the 1781 mutation, and I'm not going to go through every one obviously, but you can see what the summary there is that you've got plants there with single mutations, very few plants in the 1998 survey with double mutations. As we move through the years, we're getting more and more plants that we're finding that were resistant to Ryegrass because of double mutations, and more so in 2008. So to summarize that data, 60 to 70% of resistant plants for more years had one mutation, so we can see that even in 1998. So the mutations have always been there. 6% of resistant plants had 2 mutations in the 1998 survey, so that's these guys here, so 6%. Whereas 22 to 23 plants, resistant plants, had 2 mutations in the following 2 surveys. So we can see over time, from 98 onwards, the percentage of plants that we found that were resistant to Ryegrass because having 2 mutations increased. So that's what's happening on a large scale, is that because we're using more and more group A's, we're starting to have individual plants surviving, cross-pollinating, and bulking up those group A mutations, and then that's why you start to see herbicides not working, and even requiring higher and higher rates of, say, clethodim and butroxydim to control those plants, because we're getting a greater percentage of double mutation ryegrass plants. Okay, so let's start now talking about clethodim and butroxydim. We've had a really good sort of introduction to the group A's. So research, as I've said before, if you've got resistance to clethodim or butroxydim, it's due to group A target site resistance. Now, I've been to a lot of presentations, and I've had farmers and agronomists ask me that, oh, we got an increase in control with a DIM herbicide, so that means this is metabolic resistance. Now, that is unlikely the case. It's because the reason you get an increase in control is because as you increase the rate of those herbicides, you're starting to wean out those weaker mutations. You're killing those plants with the weaker mutations and leaving the plants with the mutation or mutations which give high levels of resistance. So that's why the increased rate. It's not due to metabolic. So if you're in a paddock and you see a ryegrass plant and it survives clethodim or butroxydim, most likely it's due to target site resistance. And as I said in the beginning, we've got a few cases of ryegrass which are really resistant to clethodim and butroxydim, but they don't possess one of the mutations we know. So we're scratching our heads with that one, and that's very interesting to find out. What could be? Is it a new mutation, or is it a new monster mechanism common to the group A's? So high rates of select improve control. Factor can sometimes improve control on select resistant ryegrass, and that depends of course on the mutations. Sometimes select is better than factor. Factor plus select mixes can offer increased control, so there's possible synergism there. But what we haven't done is I don't have any strong data with registered rates to show that the factor plus select mixes are improving control. I would like to do some more testing this year with that. The information the data I had done initially was using rates that were outside the label, and so, but they showed some positive result as well. And as we said earlier, we've touched on this, some mutations may have a fitness penalty. So, as a rule of thumb, FOPs are usually in ryegrass. Ryegrass gets resistance to FOPs. But there's a lot of cases where Achieve and Axial will control FOP-resistant ryegrass. Select is stronger than Axial, Achieve, and FOP. So there's many cases where clethodim will control Axial-resistant ryegrass, Achieve-resistant ryegrass, FOP-resistant ryegrass, and Factor can also. There are cases where Factor will control clethodim-resistant ryegrass and also the rest of the group A's, and I'll show you some information on that. So this is one trial, okay, one pot trial, where we sprayed a whole heap of special populations that I had selected. I shouldn't use the word selected because it's select and selected, that I chosen. Sprayed with Factor, 2 rates and 3 rates of clethodim. And right down the bottom, you can see SLR4 on the left. That is our susceptible population. And as you can see, we've got 100% control with all rates of those 2 herbicides. Now, if we go up the top, we can see that this population here, we've got 80% survival, or 80% control, sorry, 80% control with 300 mils of select. But in this case, this one is giving us 100% Factor 100 is giving us 75% control, 83% control, but clethodim 500 is giving us 100% control. So here's a population where clethodim is giving us better control than butroxydim. This is more common here. We've got resistance to both rates of butroxydim and clethodim, and even a leader is giving us 90% control, but we're still getting 10% surviving. So we're starting to see some different patterns occurring. Population 969 is resistant to all of these herbicides. And let's try and see some differences here. So we move here, we've got clethodim being very effective, and the higher rates of clethodim also very effective as well. You see there's a lot of different changes here. Here's one where we're getting control with factor at 180, but not with any rate of clethodim. So here's a very different response to the others. So we're seeing here that you can't always say that one herbicide is better than the other. It depends on the population. All these populations would have different mutations occurring, and that's why we're getting this huge variation in those. One of our populations down the bottom, OS93, this gives us sort of mediocre type clethodim resistance, and you can see it here. You've got quite low level resistance, 90% control with 300, factor 100. I would recommend that factor should be used at quite high rates. Using factor at 100, unless the ryegrass is very susceptible, you can actually reduce it rate. You can reduce effects. Factor 180 is a good rate to use, but obviously there are some crops you can't use it in. But this here is one trial. The next slide, this is from resistance testing that farmers have sent in, or agronomists have sent in from their farmer clients. So this has been done at different times, and I'm comparing factor 180 with select 500, and you can see the locations of where they've been collected from. But usually they go parallel. Here we've got 90% control with factor and 90% control with select. Here we've got factor working a bit better than select. Here we've got factor working, and we've got some weak resistance to select. So as you can see through there, there's quite a lot of changes, there's quite a lot of effects, but you don't always get factor controlling select resistant ryegrass. As you can see here, you've got quite a few of this intermediate type control of both of those herbicides. When you start to get, and down here where you've got high levels of resistance to clethodim, you can see factor's doing a bit better, but it's not, doing the job all the time. So there's a lot of variation between those two herbicides, but you shouldn't always assume that because you've got clethodim, you've got factor's going to work. And that is because we've really moved along in time sequence, where a lot of our ryegrass is starting to have those complex mutations, some of those stronger resistant individual mutations, or as we've seen from that mid-north information, 2 mutations or more. So I think that's a really good time to stop. That's a lot of information there.
I think so. Can you just go back to that previous slide, Pete? And I think that is the million-dollar question, isn't it? You've got all those ones up the top there where you've got sometimes factor better than select and sometimes select better than factor. So the million-dollar question, is there any way of picking when one is going to be better than the other?
Yeah, and I'll move on to that a bit later on.
Oh, okay. So we'll keep moving because we've only got 10 minutes left, but what that one says to me, though, if we just keep back at that one and tell me if I've got this right, but where you've got the high levels of resistance, so where you've got all those 40% results with clethodim down the bottom there, you sort of, there's 1 or 2 cases where factor's better, but where you've got that high level resistance, it seems that factor's pretty well gone as well. Is that right? Whereas when you get to that more moderate resistance, where you sort of 50% control with select or better, that's where there's bigger differences. Am I right there?
Absolutely. That's perfect.
Yeah. Okay. Okay.
So we've
only got a few minutes left, so we do need to plough through it, and we'll do the finish up with the questions at the end.
Okay. So commercial DNA analysis is not available at the moment. So that kind of technology can indicate to us what mutations are known. But there's multiple mechanisms occurring in plants, so you haven't always only got target site resistance. There are other mechanisms, and some we haven't discovered yet. So what information do your clients, do growers actually require? They don't really care what mutations they got. They want to know which herbicides work and at what rates, and that's again the $2 million question. So currently you can only do that with whole plant resistance testing, whether that's an in-crop testing, we're spraying different herbicides in the field, or doing pot tests with seed or with plants. That's the way. So what we're doing is treating plants the same way that farmers do, and so we look at the whole response. So this is, you know, what you can get. You got 3 rates of clethodim. You can see that response. I don't know how you could mimic or produce this result in a lab. You know, that's simple to see that. You know, if you're spraying 600, you're going to get a few plants surviving. If you're spraying at that lower rates, you're going to get more plants surviving. So that's very simple but extremely informative result. Here's trial I've done, and this one mimics that, all that other information, where the back row is all rates of clethodim. On the left is the 0, the untreated. The front is factor 80, select 500 plus factor 80, and factor 180. So if you look at the effect of select by itself and the result of, so that's this one here, and the result of factor 80 by itself, which hasn't done very much. You put both of those together, and in this particular case, because this is one population, you've got increased control, but you're still going to get survivors there. So out of this one here, the best treatment has been your factor 180 and your 1100 of clethodim, which is a very high rate. So this is one population. That's some of those monster populations that hopefully you guys haven't got. So this is information from a single resistance test, and here the farmer can see that clethodim at 500 will give you 20% survival. Intermediate, the plant surviving had intermediate type response. Clethodim at 750 gave you total control, so no survivors, whereas factor at 180 gave 10% control, and those plants were intermediate sort of in, so they had some reduction but are still quite healthy, plus some other herbicides there. So that's what a resistance test does. It can give you that kind of information. So, you know, even if you're going to use clethodim or factor in this particular paddock, you know that if you use that, you're going to wean out most of that ryegrass, but you're going to have survivors, and you should be quite active to know that you're going to do something about those guys, because if you let them set seed, the next year, next time around, it's going to be a mess. This is what I've sort of brain dumped some information and said that if you've got, I want to summarize all the group A stuff. So if you've got a test that you've only tested hoe grass, so you did a resistance test and all you tested for was hoe grass, what would that test show you? It would tell you that if you used verdict and target, it's unlikely that they would work, but the DIMS DENS may still work. If you tested only for verdict and you got resistance to verdict, then hoe grass and target would also not work, but these DIMS and DENS would work, and similarly for target. If you tested for axial only, you could assume that your FOPs wouldn't work, that achieve wouldn't work, but clethodim and factor may still work, and similar response with achieve as well. So if you've got achieve resistance, forget about your FOPs, and axial is unlikely to work as well. If you test for clethodim, forget about all your FOPs, forget about your axials and achieves. None of those are going to work, but factor may still be okay. If you test with factor and you get resistance to factor in that resistance test, forget about all your FOPs and select. You basically shot all your group A's. So that's a bit of a summary of all the resistance testing I've done over the past 10 years. That's been sort of the trends that I've seen in all the resistance testing. So this gives you an idea. When I rate some plants, triple R means very healthy, double R, which was those in the resistance test, stunting but still recovering, and single R means quite stunted, reshoots, but you know, these plants here, if left and the crop is not very competitive, will become like these triple R plants in 2 or 3 weeks. So this is still quite significant. 2 methods: quick test, that is from now onwards until September. You can collect plants out of the paddock before you spray herbicide or after you spray herbicide, get information within 4 weeks, and yeah, act accordingly. Or the seed test, which happens sort of at harvest time. And yep, we'll move on. So just
Yeah, I
to
think,
summarize.
okay, you want to just summarise it. We're just about out of time, but we might let it go another 10 minutes for anyone who wants to stay on and have some questions. But actually, can we do some questions now, Pete? I know you want to summarise it, but
Absolutely.
there's a lot of questions coming in about rate, and a lot of people saying, look, we are talking about some above label rates here and so on. And should we just be comparing, you know, the top rate of factor with the top rate of clethodim, because you know, select. So yeah, what are your comments there? Is it just a fact? Is it just that? Is it just a rate thing that, or is it a difference in the target site and so on, or a combination of both?
Yeah. The exact mutations, we don't have that information, but what is happening is that there's so much variation in those mutations out there that you don't know what you've got. Increasing the rate just weans out those weaker mutations and leave those stronger ones behind, and they're the ones that will set seed and continue the process. Now, along with just the rate, and this is some of the final slides which I may or may not get to, it's also, you know, using these herbicides at the best of their ability. So if you've got a frost coming, if you can avoid spraying them, because we know that these herbicides are not taken up, they don't work as well. So you may spray 500, but if you've got a frost the next couple of days, the herbicides aren't going to work very well. So you've got to also think about getting, you know, using the right, or using them at the right time to try and get the best activity out of them. So it's not only the rate, it's also the you know, all the other factors which cause the herbicide to work to the best of its ability as well.
Yeah. Now, one other question that came in, I mean, and I probably shouldn't have said it, but earlier on you mentioned that you're doing work with alternative herbicides and so on, and I said, what are we worried about? I mean, we are still worried, but do you want to just elaborate on that a little bit and tell us what sort of work you are doing? You know, a lot of it's probably hush-hush, but you know, how likely is it that we will get other herbicides other than what we currently have in the future, and that they'll be as good as what we've had in the past?
Okay, well, we're working with so many different herbicides that, you know, even if all those don't make it, some of those will. We've got herbicides that are selective in wheat and canola, pre-emergent herbicides, selective in wheat and canola. They're a new mode of action herbicide, and they control mouldy-resistant ryegrass. We've tested it on those. So they're totally, irrespective of what the resistance it's got, it controls them. We've got another product which is for pre and post effective in canola. So it's canola herbicide. You can use it pre, you can use it post. So with that one, we're working to try and introduce or use our existing propizamides and trophyrolins and avidexes pre-emergent and use this herbicide post-emergent. That also controls mouldy-resistant ryegrass. Yeah, there's a couple more there, but they're the two exciting ones that are quite a bit down the track, and so I think we would start seeing those guys sort of in about 2 to 3 years.
Okay, so there is some hope that there'll be some new herbicides.
Absolutely. Yeah, that's right. When I started this job in 2005, there were no new herbicides, and it's only since, you know, we all started the research, all the Australian researchers started the research into these guys, that we're now starting to see, you know, the fruits of our labour with, you know, Sakura, Boxer Gold, and now some of these guys coming onto the market in 2 or 3 years.
Okay. Yeah. So look, we are sort of at the end of our time, and so if people need to leave now, then that's fair enough, because we did advertise it for an hour. But I was going to suggest, Pete, that if we just let it run for another 10 minutes, because I think we've got a couple of good slides about the conditions and so on coming up, and if we just get to those and then maybe finish off with a couple of questions, that'd be great.
Okay, perfect. So, yeah, field failures are not always due to resistance, and you know, this is verified with a test, because there's cases where I've been in a paddock and I've said it's resistant and it's not. So you need to do something else. So how do you get the most out of these dim herbicides? First of all, you know, spraying younger plants is recommended versus the old sort of strategy. Let's wait till they all germinate and we hit them at one time. So you're hitting from one leaf to late tillering plants. And as plants get older, they don't die as easy. They get more tougher to kill. So if you spray earlier, what you're doing without recognizing is you're spraying younger weeds, but the conditions are likely to be warmer probability-wise because you're spraying earlier in the season. And under warmer conditions, dim herbicides work much better than when it gets really cold. It's just a factor of these dim herbicides is that warm conditions, you get a rapid increase in control. And also, the earlier it is, you get less chance of frost. So all those three come together, you can get improved efficacy versus leaving it another 2 or 3 weeks. If plants are stressed, any kind of stress, they don't take it up. And yeah, we've got the spray before and after frost scenario. So we've done some testing on that one. And what we found was that with clethodim here as the test herbicide on susceptible ryegrass, this is susceptible ryegrass, that if we had a frost after spraying the herbicide, we got less control, even of susceptible. So if susceptible ryegrass is growing in the presence of no frost or frost occurring before spraying the herbicide, you get much better control. But if you have a frost after, then you're starting to get reduced control. Now how about resistant ryegrass? Well, you see the rates of clethodim here quite low. We have to use higher rates to control resistant ryegrass. But again, we see the same scenario. We see that this line here is where we've sprayed ryegrass and then we've imposed a frost period on it. So we've got reduced control. So that's summarizing that if you're expecting, you get these nice sunny days, you think beautiful day to spray herbicides, but if those plants if there's been a frost, those plants are stressed and they need more than a day to recover. So that's one fact that you can get around, if possible, is to avoid those conditions.
I might just interject there, Pete, and ask you how many days do you need, before the frost, if you know what I mean, and you're safe. So with your trials there, do you know how many days after was the frost in the trial?
Yeah, these were done in a frost chamber. So what we did is we did it immediately after. So the plants were sprayed and then transported to the frost chamber. So they were imposed straight away. So we're really starting to put the plants under maximum stress. So it was immediately after spraying versus 2 or 3 days before spraying is fine. If they haven't had they've had a frost even a day right up to, and then you spray and then there's no frost period after, the herbicide takes a little while to get in. That will work a lot better than if you get that frost after spraying.
Yeah. So would you have it punted how many days you want to give it before there's a frost forecast?
So what you're saying is where you've got, if
you're spraying today and you're expecting 3 days, is that okay?
I'd give it 3 days. Yes, 3 days, and then that herbicide's got in is really starting to control that ACCase enzyme, and so 3 days after, I would expect you'd get good control.
Okay. Now, while we are doing a couple of questions, I know that Andre Sabini from Crop Care is on the line, and Kate, if it's possible, if you can maybe unmute Andre and see if he is able to make some comments. There's been quite a few questions come in about factor on its own versus select factor mixes, and I know that Crop Care had some work in this area, and if it works, if Andre can hear us and can speak, then just wonder if he might like to make a couple of comments about the work they've done.
Yeah, fantastic.
Yeah, thanks, Peter. Certainly we have done some field trials, and it was instigated from Peter Boutsalis' pot trials, but what we are finding is, I think the clethodim plus butroxydim mix that people have put into the field, it's actually reducing that unknown issue that we're getting. So you don't know what your population's got. So I look at it as though it it's an insurance policy to make sure that you put both out there. If that's not going to work, you really don't have any other options. At the moment, when an agronomist or a grower goes into a paddock, they really don't know what sort of percentage is going to be left if they run with one individual product.
So is it fair enough to say, to sum that up, Andre, if people are getting to the end of the line and really clethodim is really busted, you know, really bad poor control, the best thing to do is a clethodim butroxydim mix, so select factor mix. Whereas if people are further back down the line and select is just starting to get a bit iffy, perhaps just that might be when they might use factor on its own. Am I right there?
I'd probably say definitely for the first part, if clethodim has run out of legs, this is your final option. So, you know, you really don't have any other choices, not in that post-M side of things. And I suppose the debate with Peter and yourself would be, is I actually are promoting the use of the clethodim and factor mix prior to even having a problem. So especially in some of those marginal areas that I've got low resistance levels, I'm still promoting the mix because I see time and time again the highest rate of either herbicide should be always used, and the mix seems to be performing better than even just a single high dose of a lot of these herbicides. So I think whenever you take a tool out of the shed, and in this case, the tool for resistance management or for ryegrass control is one of those herbicides or both, you need to get the best possible control out of it. So the best possible control in our experience would be mixing the two together.
Okay, and Peter, did you want to comment about that?
Oh, look, no, I agree totally that the best strategy is to use the best tool first, and if the best tool is clethodim plus factor, then that is the best tool because we've got it. Use it, and like I said very early on, any survivors should be dealt with. But if we're going to get much better control with, say, 500 of select plus 80 of factor in a canola crop, then use that. But know that if you have any survivors, deal with them because there's nothing else you can do.
I guess the issue that we always have, and I totally agree, Andre, in principle that the mix of the two products provided that's registered is a good idea at full rates. I guess what always gets in the way is the cost, and that comes at quite a big cost over just using one product on its own, or particularly using Select on its own. And so, yeah, I guess ideally growers would use that mix. I guess what we're trying to do today is trying to work out when to bring that in, given that it is an extra cost. And I guess the answer really is, as we always talk, is the guerrilla warfare approach. Mix it up, you know. Don't be predictable. Don't always do the same thing. And so it's that mixing it up between a single shot of either product and the mix of the two, and using a range of different things. So thanks, Andre, for your comments. I don't know if you've got one or two more comments to make before we finish up.
I suppose the only other comment would be in relation to canola. So we know that the DIM chemistry or canola can be a bit sensitive to the DIM chemistry. There's certainly, it is more sensitive to butroxydim than what it is clethodim. But with that in mind, 80 grams of butroxydim is the maximum rate you can safely use in canola. We've done limited trial work with the mix of butroxydim plus clethodim, and to date we are probably comfortable that a 350 ml of clethodim plus 80 grams of factor will not significantly increase the damage that you'd get on canola. Going above those rates, however, we just don't have enough data to be 100% sure you're not going to see damage. But it's sort of like, you know, going up to that 500 mils of clethodim, you often see some damage in canola as well.
Good one. Thanks. Thanks, Andre. I think a couple of people are having trouble with sound out there, so hopefully it's all still working for most people. But thanks for your comments, and we're going to have to wind it up there. We have gone over time, but thanks very much, Peter, for your time and comments from Andre. It's been really useful. Peter, I'll just give you the opportunity to just make a couple of final comments, and then we'll sign off.
Okay. Oh, for me to make comments?
So, yeah, just make some comments.
Oh.
I've taken the, yeah, we don't have your slides up any longer, but yeah, just in the
Yep.
closing, I suppose.
Oh, okay, yeah, well, I just whip through to my conclusion, and that is, yeah, so I think because of the complexity of the issue, that resistance testing is very important, so you create a baseline of where you are, what you can use, what you can't use. So I think that's a good starting point before we're just sort of guessing where we are with respect to these herbicides. We are doing ongoing research, and this presentation to me has stirred me up to start thinking of doing some more trials to see, you know, how good is clethodim 350 plus factor 80 compared to clethodim 500, you know, as a short-term solution. So what's important? What's the activity on if you're taking a wide range of populations? But, you know, as always, you need a multi-pronged approach. So you need your pre-emergence, you need your knockdowns, the DIMs are just one component of that whole package. So we've talked only about DIM chemistry today, but that's not a solution. That is just one part. You have to use your preams, you know, rotate. Even if herbicides are working well, move away from them and use another mode of action to stop resistance occurring to any of those other mode of actions as well.
Yeah, I think that's probably a good place to finish. We've spent the whole day today talking about herbicides particularly, and we do need to always remember that herbicides are not the total answer, and really the answer is to smash that seed bank while we can and use a range of tools, and that's what WeedSmart's all about. So on that WeedSmart website we've got lots of information about the non-herbicide tools as well, and really it's the combination of doing the best with the herbicides that we've spoken about today and bringing in those non-herbicide tools to make sure that seed bank is as close to zero as possible. So I'll finish up there. Just a reminder that if you're not subscribed to the WeedSmart email list to just keep up to date with what's happening in WeedSmart, just go to the WeedSmart website and throw your name on the list if you want to keep up to date with messages out of WeedSmart and upcoming events and so on. But other than that, thank you everyone. We've gone over time. I apologise for that, but most of you have stayed on. And thank you very much, Peter, and good luck to Kate with the birth of the baby. And
yeah, good luck, Kate.
And
good luck.
We'll see you all again soon. This is the 6th webinar in the series of 6, but there will be more because they've been a great success. So